RNA. What is RNA? Ribose nucleic acid. What are the three major differences between RNA and DNA?
Deoxyribose.
Well, okay, so I guess 4. Has ribose instead of deoxyribose for sugar. What's another one? Uracil instead of thymine. Right? You guys remember that there's uracil instead of thymine in this? Okay, and I don't know if you know these other two. It is single-stranded. Okay, it's not double-stranded like DNA. Then DNA are really, really long, right? We learned that like inside one cell there's about 10 feet of DNA. Well, a piece of RNA is only about 200 nucleotides or shorter. Okay, so they're pretty short. They're pretty small things. Okay, they don't last very long. They're pretty much made, used, and then recycled. All right. We have 3 types of RNA. The first, and I'd say probably the most important type, is called mRNA. Okay, that stands for messenger. Okay? And so when we talk about this thing called transcription, this is where we turn DNA into RNA. Transcription, we're actually making mRNA. Okay, and messenger RNA sends the code to ribosomes of what proteins to make. Okay, so it's basically it's the one. That's why it's the messenger. Tells them what to make. Go ahead, Evan. Okay, so we have mRNA. The next one we have is called rRNA. Okay, so rRNA, the R stands for ribosomal. Okay, so ribosomal RNA is used to make ribosomes. Okay, so this stuff goes pretty much right to the nucleolus, the thing that makes ribosomes. It's pretty simple. That's really all you need to know about it. Okay, it goes and mixes with some proteins in the nucleolus to make ribosomes. But this one's more important. Then the third one we'll talk about, which we will also talk about later, is tRNA. Okay? And they always refer to it as transfer RNA. I think of it as more transport RNA, but transfer is the technical word. Okay? All right, and so we know that proteins are made up of what? Long chains of? Amino acids. Okay. What this does is as it reads the mRNA, as the ribosome reads the mRNA, this retrieves proper amino acid to make a protein based on mRNA. Okay? So are there any questions about RNA? Just the basic stuff about it. Okay, so I'll give you guys a quick, let's talk about transcription. Okay, so transcription, this is the process of making RNA from DNA. So you guys remember how DNA replicates, helicase comes, splits it open, then DNA polymerase reads both sides? This does the same thing, but it only reads the leading strand, so not the one where you have to put the primers on and all that. It's just the simple side, okay? But instead of DNA polymerase, we use RNA polymerase, okay? So, helicase, or DNA helicase, I should say. Helicase unzips DNA, okay? Now there are specific spots, and we'll talk about this in a second, where RNA polymerase reads, okay? RNA polymerase attaches and makes RNA strand from 5' to 3'. Okay, so again, it's just chugging along like the leading strand. I'll put that like leading strand. Okay, and then eventually it breaks off and you have a piece of RNA. Okay, breaks off when finished reading segment of DNA. Okay, so it chugs along and it reads certain parts of DNA but not other parts of DNA. Why would you only read certain parts of DNA and not other parts?
Because they're not good.
Because what?
Because they're not good.
Exactly, actually. Okay, so let's go ahead and use this black marker. All right, and then we'll use the red marker. Okay, so these things that we don't really care about, the kind of crappy parts of DNA, these are going to be the black parts, okay? These are called introns, okay? Okay, and so introns are non-coding intervening parts of DNA. So you remember how we said most mutations don't do anything, right? They're just kind of neutral. It's because a lot of our DNA is introns, okay? If you have a mutation in your intron, it's not going to affect anything because it's a non-coding portion, all right? But the stuff that we care about, the stuff that this RNA polymerase attaches to and reads, those are called an exon. Okay, so exons are the part of DNA that codes for proteins and gets turned into RNA. Okay? You guys understand introns and exons there? Basically, introns, they're just the filler stuff. Alright? The stuff that's important are the exons. Alright? That's the stuff that actually makes the meat and potatoes. The stuff that makes you, you. Okay? And so, where does transcription happen? Does anybody got a guess?
In the exons.
Nope, I mean inside of a cell. Where does it happen?
Nucleus?
The nucleus. Okay? DNA doesn't leave the nucleus unless you tear it open like we did over there. Okay? DNA stays inside the nucleus. The RNA travels outside. Okay? So transcription happens in nucleus. Translation, which is the next thing we'll talk about, which probably have enough time to talk about. Okay, that'll happen in the cytoplasm. Are there any questions about any of this?